Okay, so hopefully everybody can see that. If not, shout. Hello and welcome. It's a distinct pleasure for me to host today's cluster series seminar with a talk entitled Physiological Synchrony Across System and Species. The table of content that I've prepared for today looks rather busy, but it's really straightforward. Besides presenting results from some of our ongoing projects, in part in collaboration with other teams here at the university, I want to make some general comments and observations regarding synchrony and give you a short account of what fascinates me on the theme of physiological synchrony. And in order to exemplify that, let's start with a beautiful example of real-life physiological synchrony. The fireflies that are flashing simultaneously at night, moving from initially a chaotic, unorganized pattern to a completely ordered one. The phenomenon itself is by now well described and well understood, but it can be used to exemplify discrete steps in scientific discovery. At first, there's the observation of something so far unknown going on, that is the what. In case of firefly synchrony, it's easy. You can immediately see it, that it's happening, and you know it's real. In many other cases of physiological synchrony, you cannot. If you think of endocrine or cardiovascular activity that is now happening in coherence across people or systems, you need complex measures to gather data in the first place, and then also complex methods to analyze the data that you gather to make sure that what you think is happening really is happening. And as we'll see throughout today's talk, there's great variability across research groups in the assessment of data, and then also in the question of how best to analyze data, especially when it comes to physiological synchrony. Secondly, there's the how. Once you are certain that something is going on, you need to understand how it is happening. What are the mechanisms behind the phenomenon? In this case, the synchrony, how is that happening? How is that occurring? In the case of the fireflies, it took a little while to figure it out, but it was eventually discovered that the little creatures can phase advance their flashing in response to the flashes around them. If they observe a flash before they're ready to fire, they can engage in what you can call a power boost to increase the frequency of their own flashing until they flash in synchrony with the conspecifics around them. In case of physiological synchrony and endocrine or autonomous systems, we are much less clear on the how, although we think that sensory channels must be involved in making the synchronization happening. And then finally, there's the why. What is the purpose of the phenomenon that you think is occurring? It not always must have a purpose, but if it established itself reliably, chances are it does have a purpose. In the case of firefly synchrony, thanks to this article that is shown here by Morsef and Copeland, we now know that by firing in synchrony, the fireflies are easier to be tracked in space at night, and therefore they have an increased chance of a female finding the male who is doing the flashing. Thus, the strategy increases the reproductive success of the species. In physiological synchrony in humans, there are many more question marks related to the why. Is it aim to engage in better social integration, to increase the performance of the group, to satisfy a basic need to belong? All of these are questions that one should keep in mind when trying to understand these phenomenon and studying them across various experiments. I got first interested in synchrony by means of studying the interdependence of stress systems in humans. I still find that the interdependence of systems shares many of the methodological aspects important for studying the synchronization that also happens across organisms. In a series of studies starting in 2011, our group at the time tried to better understand how the various stress response systems in the human communicate with each other. In this first study shown here in 2011, we started looking at the crosstalk of the endocrine and the autonomic nervous systems. And this is the first method I'd like to share with you. This is the cross-correlation, allowing you to study the similarity of two time series that are shifted in time, establishing with which time shift these two are maximally coherent, using this lag in minutes that allows you to then establish the maximum correlation that can occur between systems. And using the well-known trio social stress test, which is public speaking and mental arithmetic in front of an audience, you see here the results from that experiment where we basically see the alpha amylase response, which happens very early, and then the alpha amylase response, which happens, the cortisol response, which happens with a delay. And if you now engage in a cross correlation, as shown here, you can show that the maximum coherence between the two systems occurs at somewhere around 15 minutes or so, basically showing that the original stimulation that is identical for both systems in the hypothalamus causes the alpha amylase response for the autonomic nervous system almost immediately, and then takes some 15 minutes or so until it reaches the maximum cortisol response. Now, this got us interested in investigating whether the two systems actually depend on each other. So we then engaged in a series of studies where we suppressed one system to study the effects on the other, to find a way to experimentally manipulate a system rather than just observe their response dynamics in together. So first, we suppressed the endocrine system using dexamethasone, which is a synthetic glucocorticoid. And as you can see, that is highly effective in completely preventing a cortisol response, shown here on the left panel. And interestingly, it did have a crosstalk effect on the autonomic nervous system with a higher heart rate in the group that was suppressed. So by preventing a cortisol response from happening, you basically got the other system to respond more strongly, which you can interpret as an effect in cross-talking or complementing each other in response to the stress. That was interesting. So we thought, let's see if we can do it the other way around as well. And then we engaged in a study where we used propranolol to suppress the effect of the autonomic nervous system response to stress to see whether that also had an effect on the cortisol. So first here shown the main effect of suppressing the autonomic nervous system using propranolol. You see that there's a very strong dampening or inhibition of the amylase response. And in fact, you did see a complementary increase in the cortisol response. So this is a little bit like a whack-a-mole game where you push down one and up comes the other. And of course, this made us thinking of whether this now meant that we don't see any effect on the subjective stress, which was basically the case in both studies, because as long as they are complementing each other, we are unable to really get a strong behavioral effect. So then we thought that, well, if that is the case, why don't we engage in one additional study where we suppress both the endocrine and autonomic nervous system together to then see whether that has an effect on the subjective experience of stress, which is what we did next, spearheaded by my then PhD student, Nida Ali in Montreal still. And you could show that you can actually effectively suppress both systems. So in the double suppression, after giving them dexamethasone the night before and propranolol, 80 milligrams per kilogram body weight 2 hours prior to the testing, you completely suppress the cortisol response, and you also systematically and significantly dampen the alpha amylase response and the heart rate. So basically 2 markers of the autonomic nervous system, now basically knocking down all stress response systems that you have. However, this still did not have an effect on the subjective stress experience, even though descriptively they are somewhat different. Statistically, they were not. And then also using the profile of mood states, there was no difference between the 2 groups in their mood in response to the stress, which basically is in line with other research suggesting that there's only a very weak coherence among the emotional experience, the behavior, and the physiology in response to stress. But this line now actually added a systematic manipulation for these studies so that we could show that it's not only occurring in natural variation, but also if you engage in a pharmacological suppression of the system, you still don't see a effect on the subjective stress, on the behavior, and on the emotional experience, even though the change in the physiological systems is much stronger. Okay, Now, this leads us to now engage in talking about synchronization across individuals. And the first study that opened this avenue of research, so to speak, was from Tony Buchanan and colleagues who had gathered many tests or many recurrences of TSST data, and at some point thought to group them to see whether he could actually make a link between the stress that observed that happened in the stressee, in the subject that was stressed, and the possible stress response in the observer, the person that induced the stress, that was also the experimenter in most cases in the Trier social stress test. And this would then also allow to introduce the second method, the good old general linear model, where you engage basically in group comparisons by means of statistical analysis using either ANOVAs or mixed models or t-tests, if you like, to simply see whether you can find systematic differences between groups that share or share not a physiological factor. And what Buchanan and colleagues could observe is that first, yes, there was an overall increase in the cortisol also in the experimenter. So the person that induced the stress also had an increase of cortisol over time. But more importantly, that increase seemed now to be coherent with the magnitude of the stress response in the test subject. So shown here on the y-axis is basically the change in cortisol in the experimenter and the observer. And if the person that was tested, the speaker, the stressee, if you like, had a low response, then there was also a negative change, so no response in the observer. But now as the speaker's response went to medium to high, you saw how this now led to an ever stronger change in the cortisol dynamics in the experimenter, in the observer as well. And in the highest group, this was now corresponding to also an increase in cortisol in the observer. So he named this, he coined this the empathic resonance stress response, but I think it basically goes to show that now there is elements of synchrony in the endocrine system. From there, we can move on to the parasympathetic regulation. So that is the question on whether there's also synchrony in the autonomic nervous system with its sympathetic and parasympathetic branch. And this is an example for such a study by Kathleen Gates and colleagues, who use the time-varying RSA, short for respiratory sinus arrhythmia, and that is one of the best established markers for parasympathetic nervous system activity. And this would be the next reference to the method. How do you establish the coherence or the synchrony in the parasympathetic nervous system? Kathleen Gates and colleagues suggested to use what they coined RSA linkage. And this first consists of using short epochs of time, 30 seconds windows, and then extract the frequency that you can observe during these epochs using a method called peak matched multiple windows. And then basically to engage in a correlational analysis of this RSA linkage markers that have been determined for short periods of time across subjects. And then you basically get a correlational plot where you see the strength of the correlation on the y-axis. And in this case, Gates and colleagues could show a slight but significant linkage to the marital satisfaction of the couples that they were investigating, shown here on the x-axis. The next example for physiological synchrony, not so much physiological, but here behavioral, but as an example for a method that you can also use for establishment of physiological synchrony, is the study by Fujiwara and Daibo, where they evaluated interpersonal synchrony using movement in time using a wavelet transform. And this would allow us to first establish what they did. So they basically had subjects talking to each other, and they extracted the amount of movement, the frequency and the speed, to then basically engage in a frequency band analysis of these coordinated movements. And they subjected these to a spectrum analysis of Fourier transform, which now allowed them to determine the frequency with which these movements were happening across time. And that would be our next method to quickly mention and introduce. This is the wavelet coherence, and first shown artificial data here. If you take an example where you just create some artificial data that now shows an increasing period, decreasing frequency over time, using this wavelet coherence, you can basically extract the most significant frequency band over time very accurately, which of course with artificial data works much better than it ever works with real data. But just to exemplify here, and if you now use the opposite pattern, then of course you get the opposite picture in the wavelet analysis shown here. But now importantly, if you engage in what's called the wavelet coherence, so where these two wavelet bands are coherent, then you end up with something like this. So where the two frequency bands changing over time match, you get an indication of significance, how strongly they are matched when, and you also get these little arrows that indicate the phase shift, so which band is leading the other, which one is faster than the other, until they basically are identically fast before they now move again away from each other. I think visually very beautiful to indicate where synchrony with different frequency bands occurs. And now back to the study by Fujiwara and colleagues. Using this, they basically were able to now show that there is behavioral mimicry occurring. This is the frequency analysis for one subject, the frequency analysis of another subject, and the coherence now shows where they actually match and had identical frequencies of movement over the course of the experiment. Now, this allows us to move to the next part of today's talk, which is the synchronization in groups. And there are a lot of previous studies that have looked at this and engaged in this. There's choir singing, clapping applause, which seems to synchronize in groups. And the one that I would like to quickly focus on to introduce the idea is the fire walking, which is a study by Konvalinka and colleagues that was published in PNAS in 2011. And the fire walking ritual was perhaps chosen by these authors because of the fact that it's very arousing and introduces a lot of change in the autonomic nervous system as it unfolds. And this would allow us to introduce the next method as of today's talk, which is the cross recurrence plot. That is basically a matrix where you look at the time series along both the X and the Y axes. And then the plot itself that is shown here represents every occasion at which a phase space trajectory, so a shift in the frequency band, goes through approximately the same region across the two subjects. So where they now meet the two subjects, where they engage in the same phase shift, this would now become a darkened plot here. And you can then quickly see over time whether there is a shift that occurs simultaneously in both of the subjects together or not. And there we go. This is basically the result from this study, where first here shown the heart rate change over time. You can see where the different parts of the experiments were starting. So you see the different lines, colors here, responding or referring to the different subjects participating in the experiment. So the firewalker is depicted in blue. A related spectator is the wife of the firewalker, depicted in red, and the non-related spectator in green. And you can just, with your visual inspection, already see that the related spectator is much closer to the autonomic dynamic of the person engaging in the firewalk. But using the recurrence plot here, the red marks the onset of the actual firewalking. You can see the A is the relationship in the recurrence between the firewalker and the related spectator, and the C is the firewalker with a non-related spectator, that there's much more recurrence when it actually happens, when the action is going on, when the firewalking is going on in between the two related subjects as compared to the unrelated subjects. Okay, and now we are already at the final study that I wanted to share with you today. This is in synchronization across species, or rather our attempt to see whether we can establish synchronization across species. And this is happening as part of hippotherapy, which is done by my PhD student Larissa Baer and the master student Victoria Zöppel, who has systematically contributed to this particular project. And this is a short visualization of what's happening in this study. So there's a questionnaire period in the beginning, then there's the first contact between the test subject and the horse that lasts 5 minutes. Then they engage in some brushing activity where the horse is basically now brushed by the participant. Then there's the first part of riding, which takes about 15 minutes. At the end of this riding is the lying down period on the back of the horse, which is typically perceived as the most relaxing and the most satisfying by the test subjects. And of course, it's interesting to see what possible effect that has on the heart rate of both the horse and the rider. And then there's the saying goodbye part that lasts another 5 minutes, and then another complement of questionnaires right after. We had a total of 56 women that participated in this study, 18 to 35 years old. Importantly, in this study, we systematically varied the levels of early life adversity, which we then used to form two groups with high and low amount of early life adversity to see whether that had an effect on the overall RSA activity and also the synchrony that might occur between horse and rider. And these subjects then participated in a horse-assisted therapy, which happened in cooperation with the Institut für Pferdegestützte Therapie, the Institute for Horse-Assisted Therapy, under the guidance and leadership of Dr. Annette Gomolla here in Konstanz. We were able to equip both horse and the rider with a polar heart rate sensor. There are special belts available for specifically horses that you can get, and you just clip the normal polar heart rate sensor on it so that you can now get recordings of heart rate activity over the entire session. And the analysis included RSA activity in 3-minute epochs over selected parts of the experiment. So we choose specific ones where we thought we might see the most effects. And our observations of interest, of course, relating to our hypotheses, were the RSA time course and the RSA difference between horse and rider as a proxy for synchronization, which is basically a variant of the RSA linkage method that I introduced to you earlier. So first, here is the RSA group mean over the course of the experiment. And if you just look at the riders and you summarize their RSA over the entire experiment, then RSA being a measure of activity of the parasympathetic nervous system, you can see that you start here. The initial brushing actually goes down. It's the first contact with the horse, which might also lead to some arousal. Then the riding part, it goes up. That seems to be relaxing. There's a pause in between, second riding, and then here's the lying part, lying on the back of the horse. And clearly you see the greatest RSA values associated with that, and then it's the goodbye part. If you look at the RSA differences between horse and rider as an indicator of how synchronous the parasympathetic activity is between the two, then you actually see the biggest differences in the entire group at the putzen, at the brushing of the horse, which either is not much liked by the rider or the horse, but you clearly see least synchronization there. And then you do see overall lower values for the riding and the lying down parts, suggesting that they are closer to each other as far as their RSA linkage is concerned. Now, what is really interesting for this study, I find, is if you now look at these RSA differences as a factor of the early life adversity. So if we now engage in our group split of looking at the RSA linkage for subjects with high early life adversity versus the ones with low early life adversity, you do really see the strongest differences between these two groups in the riding part, and within the riding part, then also in the lying down at the back of the horse, where the subjects without early life adversity now clearly have the lowest difference in RSA to the horse, and the subjects with early life adversity now have the greatest amount of difference, which is actually maximal for the entire experiment, suggesting that perhaps these subjects with the experience of previous trauma now might need more time to be able to enjoy the experience, to be able to relax in the presence of strangers, because of course the horses were accompanied by the therapist, but also in the company of the horse to then benefit from that. Okay, and this already brings me to the summary and conclusion for today's session. What I have been trying to show you with numerous examples from many of the studies that have been conducted in the field so far, and some of the own work that we now have contributed, is that physiological synchrony is real. It's an often observed phenomenon that occurs in different systems and in different species. And as the last study suggests, you can also find signs for it to happen across species. It has been most often studied, and as a consequence also most often observed in endocrine and autonomic systems. And it happens between diets and across diets in groups. I think physiological synchrony is also complex. There is a variety of assessment methods that I tried to just give you a glimpse of throughout this talk that coexist. And I don't think there's to date a gold standard on which method to prefer, or which method is the best, or which method is the most robust. You have a little bit the impression that everybody is basically applying what they think works best to describe the phenomenon at hand. And I think because of that, there are many open questions that need addressing at some point, which include everything from the pre-processing and just trying to wrap my head around this in the preparation for this talk. I came across interesting concept like differencing, first differencing, which is basically subtraction of the baseline, detrending, that is applied to the initial data before you even start looking at them statistically or analytically, and the initial analysis on how to extract the frequency bands. I came across very interesting methods that included the Hanning method, and then I also learned that there's a Hamming method, so the same but with a double M, which is actually not that different, versus the peak matched method that was introduced in the context of one of the studies that I showed you today. Variations in the epoch length that you use for analysis, the sliding window size that you can use, the amount of overlap, to the final statistic analysis, and keywords here, the general linear model, the RSA linkage, cross-correlation approaches, wavelet coherence, and what we didn't get into, but which certainly also exists and has its right to be used, is the intersubject correlation analysis, and something that is called cross-recurrence quantification analysis that I also showed. So coming back to the beginning of the talk, where I tried to differentiate the what from the how from the why with the example of the Firefly study, I think this is probably something where we need a lot more research and contribution also in the case of physiological synchrony, because the cause of physiological synchrony is not completely clear. There are some theories as to why they might exist, but how direct evidence is mostly lacking. There could be a vagal influence, our nervus vagus, which connects our sensory system and parasympathetic activity with our brain, has theories where you have the neurovisceral integration model or the polyvagal theory that make a strong argument that this is perhaps something that regulates parasympathetic activity and therefore RSA, but also connects us with our environment through something that some people call intuition that might play a role here. It likely involves all senses to establish and engage in what is now coined physiological synchrony. And then lastly, the purpose of the physiological synchrony, at least in humans, is less clear. And this was also back to the example of the firefly, where reproductive success, I think, is a very clear and strong argument that this is purposeful and useful. But why you engage in physiological synchrony in humans is something that people are not spending too much time on at this moment, perhaps because they are still more concerned in first figuring out what actually is happening. So what is it that they're seeing, and how is actually the best method established to robustly observe it. And once those are established, perhaps people will then also turn to the purpose, the why of the phenomenon. So existing theories and studies that I came across that try to come up with ideas on why it might be happening mention that it might increase social rapport and affiliation among the members of a group, that it could have to do with satisfying a deeply embedded motivation to belong, which evolutionary could mean that if you were expelled from your social group, that would pretty much be a death sentence. So this is something that is deeply hardwired in you to make sure that it's not happening. To increase performance, that there's actually a better performance of the group if there's something like a physiological synchrony, or to be able to detect threat, that if one of the members is clearly changing its physiology and showing signs of being highly aroused, that by means of different sensory channels, this is also communicated to alert the other members of the group so that they can respond more quickly and efficiently to what might be coming, might be an incoming threat. All right, and with that, I thank you who are still here for still being here and having listened, and of course would be happy to answer any questions that you might have.
Hi Jens, this is Markus Gröttrup speaking.
Hi Markus.
First of all, congratulations to the talk, very interesting. I have a question about slide 10. Okay. There, you treated people with dexamethasone, which is a functional analog of cortisol, in order to suppress endogenous cortisol production, which works because there's a negative feedback loop.
Right.
But how do you know that the effect on the amylase in the saliva is not also affected by dexamethasone?
That's a good question. I actually don't know. But I would hope that because the amylase is the much faster system to respond, by the time that we actually engage the subject in the stress task, which is the next day, the effects on the amylase have dissipated, and the effects on the HPA are the only ones that remain.
Okay, thank you. But have you checked that? I mean, do you have a control for that?
No, we actually don't, and I think that's where it's a great suggestion. So we should actually see what happens acutely on the amylase once you give the dexamethasone, which typically you just give the dexamethasone at night, and then you don't do anything, and you just start testing the next morning, knowing that through the prolonged negative feedback at the level of the pituitary, the HPA axis is knocked out. But it would be interesting to see, is there a direct effect on the amylase system, and how long does that take to then disappear, right? But I don't think that actually has been happening. At least I certainly haven't done it. And I would have to check the literature to see if anybody else has.
Okay, then I come up with the second question. So in slide 45, you are using people who have no or do have early life adversity experience. So how do you monitor this, and how do you distinguish these two groups? And what is the threshold or the cutoff?
Right. That's actually a popular question, because one where also a lot of variability exists between research groups and studies. So there is no gold standard. Well, I guess some people argue the gold standard is to follow them from birth and then record any adversity as it unfolds. But of course, that is typically prohibitive because of the time involved. And if you observe adversity, then you would also be ethically demanded to step in and prevent it. So what typically happens in the human studies is that you engage in retroactive assessment, and we have questionnaires that cover abuse and neglect, physical, sexual, and emotional, and then also that engage in assessing the maternal care and overprotection as additional parameters. With the combination of all these scales typically allows us to robustly identify who has had a better childhood from and separate those from who has had a worse childhood. And what also makes me believe in this method is that we do see very consistent changes in the endocrine and autonomic systems as a response to these variations in early life, suggesting that this theory about early life programming of your stress response systems really is true, and you can observe it with retroactive assessment of subjects in adult life, asking them about their childhood experience.
If no one's going to ask one, I'll ask one, Jens. Carla here. The speed dating study obviously got me interested for somebody who you know deals with the public and what they're interested in. How far along are you to completing that study? Do you have much more to go in terms of experiments?
Thank you. Good question. So I think for the moment, the data acquisition actually rests. And this also, of course, has to do with the circumstance that we're currently finding ourselves in. Yeah. Any of the group studies that we would want to do are somewhat more difficult by the situation at the moment. But what we are working on is to find the best way to actually show the results from the physiological dynamic and changes over time. So what is the best way to bring out the effect of two people liking each other and how that affects their inter RSA activity, their RSA linkage, and then also how does that look for the entire group? Do you still see patterns of RSA linkage even when they are no longer sitting in front of each other and engage with each other? So this is where I think we have more ideas to pursue and more methods to apply. The group coherence that Bernadette now has spearheaded and worked a lot on for the group stress project is something that you could, of course, also apply on the speed dating one to see whether you find patterns of group coherence in that one as well, which I think would be a very interesting next step to pursue in that study.
Okay, thank you.
Sure.
I have a question. This is Ari. Hi.
Hi, Ari.
It's really interesting talk. I have a question just in sort of thinking about the way, thinking about the current global situation in which we are all now connected on Zoom. Have you thought about doing any experiments to, or has anybody done any experiments to see how the physiological coupling between people might be affected by, you know, only having one modality and it being really sort of restricted, or restricting people's modalities in different ways, I guess.
Right. No, I think that is a fascinating question, and certainly very interesting idea to explore. We have started to think about that, trying to find ways to actually assess physiology in the first place if you are engaged in a group meeting rather than a face-to-face meeting. And of course, the face-to-face meeting is easy, right? You would simply equip everybody with the polar heart rate sensor prior to then put people together in a room where they engage in various tasks as a group. For the virtual version, you would somehow, and this is mostly logistic, right? Find ways to equip them with the polar heart rate sensor beforehand, make sure that they know how to get it on and get it working, and then assess the, start the recording before they would then also engage in a group meeting. And then you could basically just gather all the sensors after to see whether the physiology is systematically different when you have people in the room together for real, or when you have people in a Zoom meeting only being virtual together. I think that would have very clear hypotheses that you find greater synchrony if you are in the room for real, because you have all these extra modalities that now come into play that engage or will allow the people to engage in physiological synchrony. But the actual study itself is at this point missing. So I think once we have figured out on how to resolve the logistics, it's certainly something that we'll keep in mind.
Cool, thank you.
If I may add to this, I think this is actually a very good idea, because I am always so twisting my head around how much of the effects are caused by shared environment. And that way you could control for it by just allowing for visual or audio input. And so you can basically see how much similarity is just caused by similar environmental input, so to say.
Right. Totally agree. And then if you speak of environment, do you think factors that actually go beyond the person being close would also play a role, like the size of the room, or the color of the wall, or the temperature, or whether you look out into a park or out onto a wall of concrete? Is this what you mean by environment?
Yes, so basically how much is shared, and I mean, you could extend it even to topics people are talking about in your dating app, or in your dating project, or in the horse project. It could be because they're basically seeing the same scene while riding. So that may inflate the estimates of similarity just due to what is perceived, and even the movement on the horse, so to say. So I think this is a good idea, to have some control over the different environmental similarities, to basically see how much is driven just by the interaction part, and how much is driven by what people are actually looking at in the moment they're talking, or depend on the paradigm in specific.
Right, I totally agree. I think in real life, those are almost impossible to dissociate and differentiate from another. But if you do it virtually, then of course you have complete control, and you could manipulate how much they actually share in terms of what else they see, or what else they are allowed to perceive.
I have a question in that respect. How much is this perception influenced by maybe the level of distraction, or the ability to focus on a certain scene, or a task, or whatever? I could imagine, sitting in a virtual setting, people are much more easily distracted by, I don't know, a parallel activity going on in their real surrounding. Is that something, well, you obviously would have to control for that, but do you know how much stress is there? Would this add stress rather than take away, or what's it take?
I think you're actually mentioning a very important factor: the ability to engage in the present situation, the ability to let yourself be responding to the current situation. So if you have a lot of chronic stress going on in your life at the moment, there's a lot of distracting thoughts at the back of your head, you're ruminating because you have these unresolved problems that you need to deal with. All of this, you could argue, would likely not allow you to engage as much in the current activity as somebody who's free of all these thoughts and concerns and problems that they have. And I think part of this would be a possible explanation and interpretation for the results we see in the horse-assisted therapy with the subject suffering from early life adversity, that they are not able to let themselves respond to and enjoy the situation with the horse when they first engage with the horse. So this would go back to the argument that maybe this is why especially these subjects need the therapy, so they need repeat experience in the horse-rider combination to eventually be able to do that and then really enjoy in the active or benefit from the activation of their parasympathetic nervous system in that situation.
Thanks. Is that something that could be reached, for example, by just conditioning, like by repeating this the same situation over and over, and maybe getting more easily into this state of relaxation? Is there like a physical conditioning that happens?
Yeah, I think conditioning certainly comes to mind. Learning by repeat exposure, learning that the situation really is stress-free, that there is no threat to be concerned on, that nothing bad is happening to them. And of course, by repeating the experience, and every time nothing bad is happening to them, that might also then allow them to feel more safe over time and then engage in a physiological activity that is closer to what you observe in the horses.
Thanks for this super fascinating talk. So I was also like, in the past thinking about social interactions and synchrony between diets of people on a behavioral level, and I always had the problem to understand. So what is the causality of the synchronization, and how can we exclude the possibility that they just synchronize because they're doing a similar task, for example, but they don't. If I would do, for example, the Thayer stress test and measure physiology, let's say between the participant and the experimenter, this one study that you just showed, if I would completely disconnect them, I would do two different experiments and I would measure the data, wouldn't I also find synchrony, because the tasks involve similar behaviors over time, and it's like there's a synchrony. This comes from more from the task, like that they're doing the same things, or the same with the horse and the person. If I would just measure completely two different diets basically doing the same task, would I also find correlations? So I find it really hard to disentangle it, and I wonder how can we develop experimental paradigms to make sure that the synchronies are not just coming from the environment somehow, from the task demands.
Right. No, I totally agree, and this is of course an important point. And in fact, you do mention the way that this is experimentally resolved, where you can build these artificial diets and compare the synchrony you get in the artificial diet with the real diet. And of course, you would hope that if this is really a resonance of two organisms being close to another, that the real diet is superior in synchrony than the artificial ones. That is happening. So there are studies that do exactly that, and that do show the real life diet being superior to the artificial one. We also did this in a previous project, which also involved Bernadette. The problem with that is, of course, you have many possible combinations of artificial diets, right? So depending on how you choose them, you sometimes find results that are closer to the real diets or further away from them. And then it's a question of what do you take for real? Do you report all of them? Do you report the overall mean? Do you report just random? So methodologically, there are also some unanswered questions, but the basic answer is this is exactly what is being done, what you also find in the literature, where you compare the real life against the artificial diet, hoping that the real life is superior.